Hello everyone and welcome to today's live broadcast, ExpiCHO expression of HIV-1 immunogens for structured guided vaccine design, presented by Dr. Gabriel Ozorowski, Senior Research Associate at the Scripps Research Institute in the laboratory of Andrew B. Ward. I'm Susie Valdez and I will be your moderator for today's educational webcast, presented by LabRoots and sponsored by Gibco, part of Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this is an event that is interactive. We encourage you to participate by submitting as many questions as you want and at any time during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing this presentation in a slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right or the Q&A button to let us know that you're having a problem. Please join me now in welcoming Dr. Gabriel Ozorowski. I will now turn the presentation to him. Welcome, sir.
All right, thank you for the introduction, and thank you all who are tuning in today to listen. I'm going to focus my talk today on describing how we use various cell lines from Thermo, starting with the Freestyle 293 system and now expanding over the ExpiCHO system in our larger efforts in the field of HIV vaccine research. Now, the work I will be showing today as an individual is really a large collaborative effort. At the root of this, a lot of the work that will be presented is work done at 3 different institutes. Here in San Diego at the Scripps La Jolla Institute, I work in the lab of Andrew Ward. Our primary technique is electron microscopy. We also work closely with Ian Wilson, who's an X-ray crystallographer. So a lot of the structural biology work is done here at Scripps. But we collaborate closely with John Moore over at the Cornell Medical College in New York. He is an integral part in the molecular biology discovery and isolation of new immunogens. And finally, our collaborator Rogier Sanders out in Amsterdam does a lot of the immunology work, also isolation of neutralizing antibodies, topics that we'll hear about soon. So we're fortunate in this nonprofit research institute and also in the field at large for HIV vaccine research that there's a high amount of interest to get a vaccine, which with that comes a lot of labs, a lot of collaborative effort, and a lot of research funding. Now, why is there such an interest in HIV vaccines? Well, this map here estimates the amount of new cases in the year 2015, so less than 2 years ago. And globally, it's estimated that there were over 2 million new infections of HIV. And keep in mind, this is in a day and age where we have various forms of antiretroviral medications. We have different programs to help educate and to help try to prevent the spread of the disease. Even with all those policies in place, we are unfortunately seeing a rise. So, as I mentioned a few years ago, total cases in the world, about 36 million, but 2 million new cases. That equates to about 4 new cases every minute of HIV transmission. And it's estimated by multiple agencies that if we don't ramp up the efforts or change something, that we're going to see a rise of another 100 million cases in about 15 years from now. So the one way we could try to decrease this value and try to completely eradicate it is through vaccination. However, this is not a new concept. HIV research and just the description of it has been around since the early 80s. And of course, one of the first things that researchers try to do once they discover that the causative agent is a virus is to try what we've done for several other viruses, even bacterial, create a vaccine. Unfortunately, this is a very difficult model to work with, and for various reasons. I'll get to that in the next few slides. Now, here I'm showing a diagram of, let me pull out a little pointer. We have the HIV genome here, and then a cartoon representation of the virus. I mentioned that there are many antiretroviral therapies these days. The first one was AZT in the mid-90s, and that targets an enzyme called reverse transcriptase. Since then, there's several more that target that. Additionally, we have medications or drugs that target integrase and protease, two additional important aspects of the HIV replication cycle. And here quoted for rotavir, I have the estimated cost per year. And keep in mind that most patients who are on antiretrovirals, they're taking a cocktail of medications. So these are large costs. Potentially they're being covered by healthcare in some parts, but in other parts of the world, they're just inaccessible. And on top of that, all of these drugs are targeting aspects of the viral life cycle that are already after infection. So they're not preventing it. They're not eradicating the viral loads. There's some newer classes of antibodies of antiretroviral medications, and this topic will come into play a little bit later, but they are so-called fusion inhibitors. So they're targeting the initial binding of the virus to the host cell. However, these are effective at controlling viremia in HIV-positive patients, but they once again do not eradicate the virus. So what we want to do with the vaccine is target the only surface viral protein, which is called envelope. It is expressed as one long gene, GP160, or one propeptide, and that's cleaved by an endogenous enzyme called furin into two components, GP120 and GP41. Now, on top of that, we discussed there's difficulties in making a vaccine. So here are some of the elements that are a hurdle for us. First of all, HIV is a highly variable virus. It mutates very frequently. So this slide here diagrams, roughly to scale, the diversity of different viruses here. So we have at the top left corner the diversity of the global influenza strains in 1 year, 1996. So this is across the whole world, the prevalent influenza strains that were floating around. If we look below that, we see almost to the same extent just the diversity of the HIV virus in a single patient 6 years after infection. So the virus in individual patients is mutating very frequently in response to immune pressures, and also it mutates as it's spreading from patient to patient. We have a cohort of individuals in the bottom left. This is 1 year, and I don't have the total number of patients. I believe it was in the hundreds. So this is, let's say, roughly 100 patients in one small part of the world, in the big city of Amsterdam, large diversity. And then in 1 year, in one region on the right here, we have HIV circulating forms in the Congo in 1997, and in crazy diversity here. And these single letters are subtypes. So those are genetically similar strains of HIV. So the first hurdle, as I mentioned, is the great diversity of HIV. Second of all, the envelope glycoprotein on the surface that we would like to target to try to prevent infection is heavily glycosylated. It is one of the most glycosylated proteins we know in nature. These are all N-linked glycans, so they are attached to asparagine residues spread throughout the polypeptide. And in fact, it's estimated that roughly half the mass of this glycoprotein is contributed from the glycans. On the left, I have a side and top view of this glycoprotein, which assembles as a trimer. Once again, I'm going to reiterate that there's two components to it, the GP120 portion, which is accessible to the surface, and GP41, which is more hidden, and eventually it is what anchors the glycoprotein to the viral membrane. I mentioned the high genetic diversity, and not only is it diverse in individual patients elsewhere, but just globally around the world. And there seems to be pockets around the world where one particular subtype is heavily predominant over others. Now, genetically, we can break down HIV into two major groups, HIV-1 and HIV-2. HIV-2 I'm not going to talk about. It's a very small subset of populations and has only been isolated in small portions of the world. But HIV-1 is typically what we talk about when we talk about global infections. And the most prevalent pandemic group is this group M here. And that can be broken down into various subtypes listed by individual letters. Now, what I want to bring to attention is this total global breakdown here on this pie chart. Roughly half is constituted by a subtype C. And this is most prevalent in sub-Saharan Africa. Historically, however, the HIV strains that were being isolated and sequenced and that became lab-adapted strains for study were taken from patients in hospitals or research institutes in North America and Europe, which, as you see, is dominated by subtype B. So while we have several sequences and several lab-adapted strains that we can study that are subtype B, they don't necessarily correspond genetically to the heaviest hit part of the world in sub-Saharan Africa. So one of our desires is to create immunogens that would mimic the various diversity we see around the world. And this is just a reiteration of what I mentioned. The end-linked glycans on the ONV protein on an envelope constitute about 50% of the mass. And the reasoning behind this is that it has an evolutionary advantage to evade the immune system. And HIV is not the only glycoprotein, viral glycoprotein, to use glycans to shield conserved protein regions. Here on this bottom arrow, I list several others. So we have hemagglutinin from influenza, we have Ebola glycoprotein, we have HIV envelope here, and we have coronavirus spikes. So HIV envelope, the 81 is a rough estimate, but from sequence to sequence, the amount of glycans can vary anywhere from high 70s to maybe 90 per trimer. And I mentioned the evolutionary advantage of these glycans as a decoy from the immune system. This chart here looks at addition of glycans to influenza hemagglutinin, most likely as a response of evolutionary pressures due to the immune system. We go back to the late 60s. There's few end-linked glycans on the surface of hemagglutinin. And as we go through the years, more and more are added. So we believe the same rationale is happening in HIV, where these glycans offer a protective advantage for the virus. And so that is one type of evasion mechanism. Another problem, which will be very important now when we think about expressing HIV envelope as potential immunogens for vaccines, is that the protein naturally is metastable. It is able to, or it easily falls apart, where the GP120 and the so GP120 we have those kind of ovals at the top, and then the GP41 as the rectangles at the bottom. Those two components are not covalently linked to one another. So there is some shedding that occurs, some dissociation of GP120 and GP41, which leads to these GP41 stumps. You may also have trimers that are lacking certain aspects. and also trimers that did not fold properly, either due to insufficient cleavage or other factors. All of these are highly immunogenic. So in HIV-positive patients, the immune system will direct antibodies towards these stumps, towards these malformed trimers, towards these aberrant trimers here that are not fully formed. Unfortunately, these antibodies are not protective because these versions of OMF are not functional. So they will not come into play in the attachment of the virus to the host cell. Furthermore, these trimers are few and far between on a viral surface. It's estimated that each virus has maybe 7 to 14 copies of the trimer. So what we are hoping is to have antibodies against the functional, native-like trimer that is capable of infection, and hopefully by binding antibodies, it prevents binding of the virus to the host cell. Now, for a while, this was more of a dream or an ideal than anything, but over the past decade, we've, as a community, have been isolating more and more antibodies from HIV individuals who we deem elite controllers. These are individuals who are HIV positive, but their prognosis does not progress towards AIDS, and their viral loads are relatively low. So something naturally in their blood, in their body, is able to suppress the viremia. It doesn't eradicate the virus, but it prevents the need even for antiretroviral therapy or intervention at the time. And what's been discovered is that a subset of patients, maybe 25%, produce what are called broadly neutralizing antibodies. These are antibodies that are not only protective against that specific strain of HIV that infected that individual, but on a larger panel, it prevents in vivo infection of other genetically diverse strains. So because these patients naturally produce such antibodies, we feel that there should be a way to recapitulate this in the form of a vaccine in healthy individuals, and hopefully they will have circulating broadly neutralizing antibodies that will prevent infection if they are ever exposed to the virus. This is not a fully comprehensive list of all the antibodies, but I list them here on this chart in terms of coverage, so percent viruses on a panel that would be neutralized or infection would be prevented if you incubate with this antibody, and also the potency on the x-axis. I mentioned early on that our lab primarily focuses on electron microscopy. One of our advantages of our tools is that we're able to directly look to see where on the surface of these envelope trimers these antibodies bind and map these epitopes. As we've discovered over the years here, there's very few surfaces that are left untargeted now by these antibodies. So before we thought maybe only the most conserved ones would be the targets, but we're learning that that's not always the rule. Now, with electron microscopy, with low-resolution models such as the ones you see to the right, those are quick experiments to do relatively easy, and they help us determine the epitopes. They don't give us the amount of detail we need to figure out which amino acids are responsible, but they're a good starting point. Then what we do in our lab is we move on to cryo-electron microscopy, which is a high-resolution technique, and in recent years it's overlapping with X-ray crystallography for the amount of detail and amount of resolution we're able to get. And just as a proof of concept, this is from a study years back. If you take these broadly neutralizing antibodies and you put them into macaques or monkeys and then try to infect them with a viral or HIV version, a hybrid that would infect monkeys, you see that there's protection of these antibodies. So at high doses, kind of medium doses, and even at low doses, several animals respond by not actually becoming infected with the virus. If we use a control, this is, I believe, a dengue antibody, which does not offer any protection against HIV, all the animals do get infected. So there is number of studies that demonstrate these broadly neutralizing antibodies are very effective. And they could even be used as an alternative to small molecule antiretrovirals, which some groups are working on, but that's independent of our research. So just to recap, we've talked about diversity, genetic diversity of HIV being a big hurdle. We've talked about the glycosylation aspect, and I've alluded to the metastability as another viral decoy. And this is the problem here, described at the top. This dissociation of the components of the trimer, GP120 here, GP41, leaves open these surfaces that are normally packed against one another. They're not accessible to the immune system. Now all of a sudden, they're available to the immune system. They're highly immunogenic. You'll get a great immune response. You'll get antibodies against them, but they once again won't be protective because they don't bind to the version of the trimer that is necessary for infecting new cells. Now, of course, this is a big protein engineering problem here. If our natural protein has fallen apart, it's going to be hard to put this into a vaccine. What our collaborators, specifically starting with John Moore over in New York, going back to the early 2000s, they came up with a clever idea on how to make this into a form that we can actually use. Here's a schematic of the envelope peptide. The GP120 is this cyan and orange region here onto the left. There's constant regions listed as C. There's variable regions. There's 5 variable regions listed as B. And beyond that, we have GP41 here. We have in red this membrane proximal external region. And we have this transmembrane domain and a C-terminal domain, which is thought to be on the inside of the virus. So we're dealing with a number of issues. First, this detachment of GP120 from GP41. So let me clear this. Our collaborators were able to address this by introducing a disulfide bond that links GP120 to GP41. And the great thing about this disulfide bond is it still allows for the cleavage by the furin enzyme to occur. We find in our studies, our structural studies, that if the cleavage does not occur, these trimers do not fold into their native-like state that is capable of infection. So we have a disulfide. The two won't separate from each other because they're covalently linked, but the cleavage still occurs for proper folding. Second, they introduced a simple isoleucine proline mutation in the GP41 region, and this prevents the helices in that region, which during the fusion process do elongate. We want to keep them in their pre-fusion state. So this proline adds essentially a kink that keeps the energy minimum in the pre-fusion state. And finally, they've been able to truncate at this residue 664 here. So this is before all of the kind of hydrophobic membrane proximal external region and transmembrane domains. So we're able to solubilize these. Once again, to recap, this is a crystal structure here of these engineered envelope trimers that we use. SOSIP is the term that you may hear throughout this presentation, and that signifies the SOS linkage, so the disulfide, and then the I to P mutation. So these SOSIP trimers, once again, we have GP120, GP41 intact. They're covalently linked, so we shouldn't see that shedding that the virus naturally does. And we want to ensure that everything else is in its natural state. These V1, V2 loops are kind of at the apex of the trimer. There's been many neutralizing antibodies discovered that target that region, so we want to make sure it's presented properly. We also want to occlude this V3 loop. It's tucked in red here underneath the V1 and V2. It tends to be immunodominant and immunogenic. However, those antibodies generally do not offer protection against infection. So we want to make sure that's nicely sequestered in there. And finally, we keep good tabs on the glycans want to present them as naturally as possible. And as a proof of concept, we've done several immunization studies throughout the years to demonstrate that these SOSIP trimers, compared to other designs which have removed that cleavage site, are superior in terms of immune response. So the three here on this chart, this is the amount, the titers of neutralization or antibodies that are able to neutralize the parent virus that these immunogens are based on. The three with the largest success were the SOSIP designs from two different strains, B41, BG505, those are from subtype A, subtype B, and all these other designs that rely on deleting the cleavage site or introducing a fold-on trimerization domain did not offer the same level of protection. So our group, and in collaboration with the others mentioned, we have this iterative process. You know, at the core, because we are an electron microscopy lab, we are able to evaluate these trimers by electron microscopy. So anything we're expressing, and from now on, most of the work I will show will be from the Freestyle 293 system. Later, we'll compare it to recent efforts with ExpiCHO. So what we express in-house or via collaborators in the 293 system, we're able to examine by electron microscopy. But even before that, you know, we do size exclusion chromatography to see the relative ratio of trimer formation versus dimer and monomer, protein stability, DSC, nano-DSF as techniques, and then, of course, antigenic profiles. What we care about here is that the proteins we express, that they're capable of binding to broadly neutralizing antibodies, but they limit the binding of non-neutralizing antibodies. And finally, before any kind of clinical studies or animal studies, we like to do some types of stress tests to see the stability of these trimers under various conditions. This is a very brief overview of the assessment we quickly do by electron microscopy. Specifically, the technique is negative stain electron microscopy. We have these small copper grids on which we've deposited a thin layer of nitrocellulose, so that's in gray here. That's also coated with carbon to help absorb proteins. We put our protein sample down, we add a heavy metal stain, we blot it off, and we can store it in these grid boxes until we're ready to collect data. When we collect data, this is a representative micrograph. So the way this negative stain works is that it will prevent the transmission of electrons through this grid everywhere where there's stain deposited, but when you have protein particles, they tend to occlude that stain, so the electron beam transmits through them and the particles appear as white. So even just looking at this micrograph, if you squint your eyes, you'll see protein particles that appear to have a trimeric shape to them. We're able to do some automated particle picking, and then we can extract those particles. And if you look at just the individual particles, there tends to be different levels of noise, signal to noise. Some of these do not, even represent the trimers yet, but through computational algorithms, we're able to group these together into ones that share similar features, rotate them, translate them, and create what we call class averages. So here we have, more defined, because in each of these subsets, you're averaging maybe between 50 and 100 of these particles. Now, why we do this, through our experience, we've noticed that there is a correlation between trimers, expressed trimers or engineered trimers that offer the correct antigenic profile, and then furthermore a good immune response, versus those that lack that. We find that the trimers that work best have this kind of closed conformation, which we call native-like. There's no sign of the trimer falling apart. It looks like all three of these GP120s from this top view are interacting with one another. We also see some degree of flexibility. Overall, the central density here is still trimeric, but these extra densities on the side may suggest that some of the domains are moving around. And the non-native trimers tend to look like this. You may have, the 3 GP120s, but they're not associated with one another. They're splayed out, but because they're covalently attached to that GP41, they're still tethered to it, but do not adopt this closed compact state here. And on top of that, as I mentioned, we've been able to look at various antibodies bound to these trimers. Coupled with this, through other collaborators, specifically Max Crispin at Oxford University, they use mass spectrometry to really analyze the glycan profile of these trimers. And the main take-home message here is these are expressed in mammalian cells or human cells if we're using HEC293F, or mammalian hamster if we're using the CHO system. So we would expect from a glycan processing standpoint that all of the end-linked glycans should adopt a complex glycan glycoform. What is unique about HIV is that there are so many glycans, and in regions that are so dense, so one next to each other, that they are actually preventing some of these glycan processing enzymes from getting in there and having access. So what we find is that there's a high degree of glycans that still are the high mannose or oligomannose premature form. Most of those are located in the GP120 region and are shown here in green. We have more exposed glycans that don't have too many neighboring ones that are processed into complex ones in pink. And then in orange are hybrid ones where they're not, still haven't fully gone downstream. And on top of that, I already alluded to the fact that cleavage is important for proper trimer formation, and we've been able to show that by electron microscopy. Some of these, the top panel here is our SOSIP design where we allow cleavage to occur, but we have that engineered disulfide and the I to P mutation. And if we start removing some of those regions, or if we start putting in just the SEKS removes the cleavage site, you see more and more of these phenotypes that are aberrant trimers splayed open. Additionally, we feel that we should be preserving the glycan forms as well as possible to that of the native virus. But if we do not engineer our proteins the correct way, that has a strong influence on the glycosylation processing. And the story here is, once again, if we take our SOSIP design, those closed compact trimers, we get a glycoform that is predominantly high mannose. If we instead look at the trimers that are splayed open and just tethered by that disulfide bond, we see more and more glycan processing, more of the complex glycans, which do not necessarily represent what is on the surface of the virus, and therefore does not represent what the immune system sees. And on top of that, our purification scheme for these trimers varies from construct to construct, and this will come into play very soon when we talk about ExpiCHO. But there's 3 different purification schemes I would like to address here. There's 2G12. This was the original antibody we used for purifying these trimers. It binds to a high mannose glycan patch on the surface of GP120. So this antibody, isolated from a human individual, actually targets the glycans because it's so different from human glycosylation due to the density and the immature form of the glycans. We're able to use this to purify the trimers. It would be a more selective type of lectin purification, so to say, because it's specific against HIV, but it does not discriminate between dimer, monomer, trimer. We also use PGT145, which is an antibody that binds the apex, and it specifically binds to trimers. So we can select for the correct oligomeric state there. And PGT151 also binds to trimers, but it will it binds at the interface between GP120 and GP41, and it will only bind if that furin cleavage has occurred. So we have some of these are highly selective, and we use them depending on the strain we're expressing, because not all of them are amenable with each form. Some of them rely on neighboring glycans, so we'll come back to this in a little bit. And the choice of the choice of purification, especially the affinity step, does matter. Here we published a paper where we show, if we're using for some trickier SOSIP constructs, if we choose the less selective 2G12, we get a mixture of maybe 50% of the trimers appear native-like, the rest do not. If we instead choose the PGT-145, which only selects for trimers, we get close to 100%. And if we choose less stringent techniques such as a his tag and nickel NTA, SEC, most of the proteins look like aberrant trimers. I'm going to quickly go through these slides because they're not as pertinent here, but they do tie into the story that we want to reproduce or recapitulate the proper glycosylation as much as we can, because some of the glycans are highly consistent in both their presence and also the glycoform. And they are the target of certain broadly neutralizing antibodies. So therefore, it's important that we keep the right glycoform there. Hopefully, in a naive individual, they will be able to produce antibodies that target similar epitope that relies on these neighboring glycans. And others are variable. As I mentioned, over the course of infection, the virus mutates quite quickly due to immune pressures. So what the immune system initially sees will be different from what it sees 2 years later. This is something we keep in mind in our immunization studies because the germline antibodies, or the precursors to these broadly neutralizing antibodies, probably bound to a version of the trimer that is different from the one that the mature broadly neutralizing antibody binds to. So this gives us this idea that we should investigate sequential immunizations as long as we have information, longitudinal studies of the viral evolution. It is something we can do. So now actually going into what we need, how we're doing it, and new things we're trying. We need for our studies, not just in our lab, but in our collaborations, recombinant antibodies. I've alluded a lot to studying broadly neutralizing antibodies. So we need those both for our assays to validate our proteins or our immunogens, but we also need them for our structural studies. Both neutralizing and non-neutralizing antibodies, because we want to screen against both, and both IgG and fragment antigen binding or Fab forms. The Fab forms tend to be more amenable to structural determination because there's less, you remove the flexibility of the hinge region with the FC portion. And of course, the biggest part of our studies are these soluble trimers. We use untagged versions for immunizations in animals, but we also create labeled ones that we can bind to our probes, whether we're doing SPR, ELISA, or Octet. What are requirements? Okay, so going back, the requirements prior to this were, we have stable proteins, so we're able to do that through our engineering scheme. We want native glycosylation, so we are adamant about using mammalian cells for this work, and the quantities required, are important to factor in here. So if we just talk about antibodies, because this is the easier aspect of it, antibodies or Fabs. If we want to do our octet red, biolayer interferometry system, we generally expect that we would need about 100 micrograms of IgG or Fab to do one 96-well based assay. So these are reasonable quantities, relatively easy to obtain regardless of the system we're using. For structural determination, we might require up to 1 milligram, in this case of Fab. So once again, these are not, extremely difficult quantities to obtain. using the Freestyle 293F system, which we were using up to this point, and we'll talk about in a little bit why we're switching. But for a more elaborate experiments, there's some animal studies where we would like to study immune trafficking throughout of the immunogen to germinal centers or the immune system. And for that, we require intact IgG. And we're estimating that for a full study in monkeys, considering all the groups, we might need close to 1 gram of IgG. This is a large amount. So how do we express these things in our lab? First of all, we transfect into either HEC293F or ExpiCHO. We've been using 2-to-1 ratios of heavy chain to light chain, either for the IgG or the Fab. These were values that seemed to work well for our collaborators initially. To be honest, we haven't played around with it much more beyond that because of the yields we were getting. We were happy with them. And like I said, either HEC293F, which we've typically been expressing for 6 days prior to harvesting. Also, when we started doing this, we were using PEI. It was, for us, a cheaper alternative than the 293 fectin system, and our yields were at least 50% of what we could obtain with 293 fectin. So at that point, it was cost effective to switch. Now we're also experimenting with the ExpiCHO system. For antibodies, we're following the max titer protocol and harvesting up to 2 weeks post-transfection. We purify these mostly by protein A resin or some type of mAb select if it's an IgG, using our FPLC systems in the lab. If it's a Fab, we have kappa select or lambda select columns against the kappa or lambda light chains. And generally, we're also purifying those over a mono-S cation exchange column in order to remove any light chain dimers that may have formed. So how do these compare? Like I mentioned, originally we were using the HEC293F system with PEI. I would say on average, and it varies from antibody to antibody, and I will also include that one of the variations is probably due to different vectors being used. Oftentimes we're not designing the genes here or popping them into vectors. Another lab has already done that, so they send us the plasmid, and we kind of assume that it's going to be good enough for what we want. So on average, per liter of culture, we were getting about 10 milligrams of recombinant IgG. At best, maybe 25. For Fab, generally less. We think it's more of a expression issue of the light chain and the heavy chain binding together without having that FC portion to help out. But nonetheless, we could probably get about 5 milligrams per liter. Now, we're using the ExpiCHO system. So ExpiCHO system, following all the reagents and the manufacturer's protocol, on average, we're getting about 310 milligrams of IgG per liter. So that's an increase anywhere from 10 to 35 times what we were getting with the 293F system. In some cases, with 1 liter, we're almost hitting that 1 gram mark that we were estimating we might need for some of these fancier immunization experiments. And it's funny to think, maybe a year ago, less than a year ago, we were thinking about, something like that we would have to outsource, do elsewhere. Now it's something we can easily do in our lab just by switching over to this expression system. For Fabs, similar story. In general, about 10 times higher yields than what we're seeing. There's been some experimentation trying to use the pcDNA 3.4 vector from Thermo to see if that can increase our yields even more. Our initial experience is that we might get another 20% boost from it in terms of total yield. But not in all cases did we examine looking at codon optimization for the specific cell lines we're using. So there's still a good amount of work that can go in here. And the Fab yields do suffer after mono-S. There does seem to be some precipitation, but that's independent of the cell line used. So In general, for any type of antibody, whether it's Fab or intact IgG, we are extremely happy switching over to this platform because we're able to make at least 10 times more, and usually more than that. So what can we do with this increase in the expression using ExpiCHO? Well, I'm estimating here that if we take an average of 310 milligrams of IgG from a liter transfection, that's 3,100 octet experiments. So for one antibody, we could probably make a stock, freeze it, and for the next decade or so in our lab, anyone who wants to study the binding of that or the kinetics of that specific antibody to any new trimers made can be using the same stock without having to express it again. Obviously, we don't need such large quantities for one specific antibody, but here diversity matters. There are several different broadly neutralizing antibodies, several non-neutralizing antibodies. So if we can cut this, do 100 milliliter transfections, get 10 times more diversity, that's great. Also, these affinity columns that I mentioned before, the 2G12, PGT145, PGT151 that we use to purify trimers, we generally couple about 20 milligrams to Cephalus 4B. So we can generate 15 new columns that we use in a gravity flow and peristaltic pump format. This is great. These columns have high binding capacity for about 2 years, and then they need to be replaced. This allows us, with one expression to literally give everyone in the lab their own column if we wanted to go that route. And like I said, it's enough for one of these immune trafficking experiments in the CACs that we're proposing. Now, with the amount of Fab we can get, 30 milligrams, let's say on average, that's 30 cryo-EM structures. 30 cryo-EM structures of the same antibody might be redundant. So let's, express smaller volumes and make 30 Fabs. Now we have 30 unique cryo-EM structures, which is great for a lab like us that relies on publishing new and novel complexes. About 300 octet experiments. And what I like to iterate is that we're getting these large boosts in yield, but we're also saving costs. So I've estimated that using the ExpiCHO system and the average yields we're getting for IgG Fab expression, it is 1/3 the cost of if we did this with HEC-293F plus PEI. Partly due to consumables, the flasks, the DNA, everything going into it. And this does not include labor costs. It's actually much faster to purify these when you do smaller scale and still get the same amount of material. So we'll tie everything up at the end, but for antibodies, we are very happy in our experience, and not just our lab, but John Moore's lab at Cornell Medical College, and also Rahir Sanders' lab in Amsterdam. They've all been working with this ExpiCHO system now after our success. So it is our go-to one for antibodies related to HIV research. Now, SOSIP is trickier. We mentioned metastability, glycosylation, all of these other factors. What we need for a typical octet experiment is anywhere from 200 micrograms to a full milligram of this SOSIP trimer. For structural determination, roughly same amounts. And typical immunization experiments for one specific type, genotype of a SOSIP trimer, anywhere up to 5 milligrams, depending on how many groups of animals we're using and what types of animals. These are also expressed traditionally in either HEC-293F system or the ExpiCHO. And here we're transfecting 4:1 ratios of the SOSIP plasmid, but also a DNA plasmid encoding for the furin protein. I mentioned this is endogenous, so it's naturally expressed in human cells, but due to the overexpression of SOSIP, it can't keep up. So if we run gels afterwards, SDS-PA gels, we see that cleavage is not efficiently occurring between GP120 and GP41 unless we co-transfect with additional furin. For 293F, similar process to what we were doing for the IgGs, 6 days post-transfection harvesting. For ExpiCHO, we've tried a couple different things, generally following standard protocol. I've tried the high titer one. It didn't help with this aspect. And we generally harvest 7 days post-transfection, upwards of 10. And our collaborator finds that adding a second feed on day 5, even if we're not following the max or high titer protocols, helps get us higher yields at the end. We're purifying these over the 3 anti-HIV affinity columns and size exclusion chromatography to isolate just pure trimer, no aggregate, no dimer monomer. So here's a comparison now from how we were doing it, how it is in ExpiCHO. I would say that there's a modest improvement in HEC-293F. On average, we're getting maybe 0.75 or 750 micrograms per liter of transfection. Now, we don't measure directly total expression transcription levels. What we think is occurring is that we are getting high levels of expression, but only a small subset is actually forming into the trimers that we are interested in isolating. If we go to ExpiCHO, we can get maybe upwards of 5 milligrams per liter using the system. So we see a fold increase 5 to 7 times of what we were getting using 293F. Now, I mentioned the lower yields, the complexity of this protein, heavily glycosylated, that could be just a natural issue, and it's such a unique protein that it's hard to compare to other things in the field. We've had some anomalies at first observed by our collaborators. Now, if they chose to use this PGT-145 column, which binds at the top of the trimer, they were noticing lower yields if they put ExpiCHO material over this column compared to 2G12. We haven't seen this before if we use CHO stable cell lines, so these are cell lines that are stably transfected with the SOSIP gene. So it seems to be something different in the ExpiCHO system. Our hypothesis has been that the expression levels are so fast and so rapid that maybe we aren't getting proper glycan processing for some critical sugars that are involved in PGT-145 binding. But our solution to this has been to either increase the amount of resin or do batch binding or a double pass over columns. So collaborator was able to split a 300 mil ExpiCHO transfection into 2. One he did over the 2G12 column, single pass. One he did over a 145 column double pass, which was packed with more beads. Final yields are about the same. So there does seem to be something that is affecting the binding, possibly the increasing off rates of the SOSIP trimer to this PGT-145 antibody if we are expressing an ExpiCHO. However, our workaround is to increase the amount of resin or batch binding versus regulated flow overnight. Now, I mentioned the modest increase 5 to 7 times. The way I would see this too is, I keep mentioning we're interested in diversity. The HIV virus has a high amount of diversity. We're trying to create a vaccine that would be useful in different parts of the world that tend to be infected by genetically diverse genotypes of HIV to other parts. So it's imperative that we test a lot of different SOSIPs based on a lot of different designs. And in our incubators, our SU2 incubators, generally with HEC-293F, we're doing 1 liter transfections in the 2 liter flasks, and we're able to fit roughly 8 at one time. Also, loading a liter over these columns at slow volume takes multiple days. With ExpiCHO, we can reasonably, cut down our volume by 1/10 and do 32 constructs at the same time. So this is advantageous if we want to quickly screen through different designs by octet and other studies. And it would still give us enough potentially to even try some EM work. Let me clear this. So what I've estimated too is because of less consumables, we're able to test maybe 4 times as many variables or different SOSIP trimers using ExpiCHO system for about a cost that's half as much as, for a cost that's less than 2 times the cost of equivalent 293F transfection. So it's a little more expensive, but you can get more done at a time. So once again, the goal here is to increase the diversity of potential immunogens. It's better for us to screen more in a 1-month period than have to do that over a 6-month period. It's a fast-moving field. So this is where I see an advantage of using the system. So finally, I'd like to conclude in terms of antibody production. We've essentially completely switched over to ExpiCHO. I would highly recommend it. It saves money, and you get more out of it. And different types of antibodies, whether it's IgG or Fab, ones that express worse, and others, we've had success with all of them. So that's the greatest aspect of this. And for SOSIP expression, as I mentioned, it's going to be case by case. This is a difficult protein to work with, but if we're interested in diversity over a shorter period of time, it is a feasible way of us doing that. So I'd like to quickly thank members of my lab, my mentor, Andrew Ward, several other key players in our research, and the other labs that I mentioned, the Wilson Lab, Moore Lab, Sanders Lab. Our funding comes from multiple sources, NIH grants, Bill and Melinda Gates Foundation, IAVI, and IAID, all working towards HIV vaccine research. And finally, I'd like to give a shout out to Issa Moody and Rick Bossi for encouraging me to present my findings today to the audience. So I will now take any questions. So thank you.
Thank you, Dr. Ozorowski, for that informative presentation and your important research. We will now start the live Q&A portion of the webinar. If you have any questions to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your questions into the box that appear on that screen, and then click the send button. We'll answer as many questions as we have time for. So let's look and check out our questions. One audience member would like to know, what is the next step in optimizing the ExpiCHO expression of HIV-1 trimers?
So, sorry, okay, I'm back. So I think the important aspect to really look at is to go back to the genetics, the gene design. I mentioned that several times we're getting plasmids from other collaborators, but I think it's critical if we want to boost the yields even more beyond what we've initially seen to better design our vectors, choose the best expression vectors for the correct cell lines, so whether we're doing ExpiCHO or 293F, and codon optimization, all of that. So I think that's the next step, but also taking another look at total amount of DNA and DNA plasmid ratios during transfection. I mentioned we're in a fast-moving field. A lot of times we're excited to try these new methodologies, new techniques, new reagents, and we jump into it. We have some success. We can definitely optimize it, and I think we just need to go back to the drawing board, have a few people devoted to testing those variables.
Thank you for your answer. Here's another. How do trimers produced in CHO versus HEC293F compare in terms of protein stability and immunogenicity?
Yeah, that's an important question. We are just beginning to look at the immunogenicity of ExpiCHO produced trimers, but based on our previous experience by expressing in CHO stable cell lines, we did not see any difference in controlled studies in macaques, whether we produced the same trimer in the 293F platform versus CHO. both responded well in terms of the immune response. Second, we generally do not see a difference in the thermal stability. If we run DSC, nano-DSF, roughly the same amount. And on top of that, the antigenicity profiles seem to be very comparable. I would like to finally add that we are collaborating to have one of these early designs, or one of the initial SOSIP designs, large-scale production, GMP scale, for human clinical trials, and that type of work will be expressed in CHO cell lines. So we have confidence that the two are interchangeable for our work.
Thank you for those answers. Unfortunately, we have no more time and for questions, but additional questions will be answered and published on Thermo Fisher Scientific's website. You will receive an email notification when answers are available. I'd like to take the time to thank LabRoots and Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available on demand viewing through April 2018. You'll receive an email from LabRoots letting you know that the webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's event. That's all for now. Enjoy the rest of the conference. Hope to see you again. Goodbye.